Hello everyone, my name is Chila Askar. I'm from class 12-3. In this video, I will briefly explain a biology topic from the chapter Protein Synthesis. What do you guys think if you heard that two words, protein and synthesis? Do you know what the meaning of protein synthesis? Before we begin, I would like to express my sincere gratitude to Mrs. Ismi Sanjayani as my biology teacher and also my beloved homeroom teacher for giving me the chance to make this video as a school assignment for this semester. Without further ado, let's move to the video. Enjoy watching! Protein synthesis is the process of protein formation in the body carried out by RNA on the command or code from the DNA. Protein synthesis takes place in the cell nucleus and in the ribosomes. Do you still remember about ribosomes? Ribosomes are cell organelles that are small and dense and functions as a place for protein synthesis. It is also called synthesis. It must require raw materials. Well, the raw materials in protein synthesis is amino acid. The number of amino acids involved in the protein synthesis is 20. However, the amino acid sequence is fully encoded by DNA. The end result of this protein synthesis is a formation of functional proteins such as enzymes, keratin, hormones, and hemoglobin. And then, what is the mechanism like? The mechanism of protein synthesis consists of two stages, namely transcriptions and translations. Transcriptions is the process of printing RNA by DNA. The formed RNA is divided into three, namely tRNA, mRNA, and rRNA. The order of amino acids will be determined by the nitrogen bases attached to the mRNA chain. The order of the transcription process is as follows. The helical bond of DNA will be opened by the RNA polymerase enzymes, and a copy of the genetic information from the DNA is formed. The place for RNA polymerase to attach to DNA is called promoter. The promoter functions to show the place where the transcriptions begin and determine the DNA template from the two existing chains. This process is called initiation. Two DNA chains consisting of a chain of mold or sense and complement or antisense begin to separate. For the promoter to stick to the mold chain, RNA begins to be formed by RNA polymerase from the starting point of the promoter and moves continuously along the DNA mold chain. As a result, the DNA helix opens sequentially along 10 until 20 nitrogen bases. These nucleotides are assembled from the directions of 5 until 3. This process is called elongation. The nitrogen base formed in RNA is a nitrogen-based complement in the DNA sense chain, which is as follows: T based on DNA for A print on RNA, C based on DNA for G print on RNA, A based on DNA to U print on RNA, and the last G based on DNA for C print on RNA. After RNA polymerase transcribes the terminator DNA, the transcription process will stop. This process is called termination. Then, RNA will detect from RNA polymerase that causes the helical DNA to close again. mRNA produced from the transcription stage will exit to the cell nucleus to the ribosomes. Translations is the translations of RNA genetic code into an amino acid sequence. At this stage, polypeptides will be synthesized using the genetic code of mRNA in the ribosome by tRNA with the help of the tRNA amino acid synthetase enzyme. Translations also consist of three processes such as transcriptions, namely initiations, elongations, and terminations. The order in the translations process is as follows. The mRNA molecule binds to a small ribosomal subunit at the 5' end. Translations start from the start codon, which is the AUG in the mRNA, then tRNA as an initiator that contains UAC anticodons will carry the amino acid methionine. The amino acid will be attached to the AUG codon. Remember, methionine is the initial amino acid in protein synthesis. Next, other amino acids will be added by the tRNA amino acid enzyme until a complete polypeptide chain is produced. Such translations will continue until the stop codons appear, namely UAA, UGA, or UAG. If one of these codons appears, the translations process will stop. The polypeptide that is formed will detach from the ribosomes. Briefly, protein synthesis can be written as follows. DNA transcribed in the cell nucleus to form RNA to mRNA leave the cell nucleus to the ribosome by carrying the codon. tRNA will carry amino acids according to the codon to the ribosomes. It's called translation. Amino acids are arranged according to the genetic code in the codon. The compounds are formed polypeptide or protein. Hopefully what I explained can be useful. Sorry if there are any mistakes in the delivery of the material. Thank you for watching and see you again. Bye bye.